Good afternoon. My name is Brittany Littlejohn, and I'm an assistant professor here within the Department of Animal Science and Division of Agriculture in the University of Arkansas system. And today I'm going to be visiting with you a little bit about some research that we have in collaboration with the Livestock and Forestry Research Station there in Batesville, Arkansas. This research evaluates the influence of prenatal exposure to toxic fescue on health and performance of weaned beef calves. Now, this project focuses on prenatal programming, and prenatal programming is defined as the fetal response to a specific challenge during a critical developmental window that actually alters the trajectory of development of that fetus. And so if you look at our bovine fetal development timeline here, we can see that different physiological systems are coming online in that developing fetus at different times in gestation. So if the dam of that offspring experiences some kind of event at a certain time point in gestation, so maybe that's a change in nutrition, whether positive or negative, maybe that's some kind of stressful event, that has the ability to actually impact the dam, thereby impacting the environment of the fetus, and thereby impacting these developing systems that are coming online in the fetus itself at different times during gestation. And so this is going to actually have the ability to alter the outcome of that offspring in a long-lasting way throughout that animal's life. And so that's when it becomes relevant to us, for example, as beef cattle producers. So what can cause this prenatal programming event that might be caused by the experiences of the dam altering the fetal environment? How is it actually going to alter and program the fetus itself? So we largely know that one of the primary contributing factors to prenatal programming is something called epigenetics. And epigenetic modifications, such as DNA methylation, which is the type that we look at in our research, that these are going to cause changes in the DNA packaging to lead to alterations in gene activity. So this might result in activation of genes or turning genes on, or silencing of genes and turning genes off. And so really to understand epigenetics, we need to kind of take a step back and better understand what is the function of epigenetics. Now, epigenetic modifications primarily exist to regulate cell-specific functions. So we know, so many of you have probably heard, every cell in the human body, the animal's body, is going to have the same underlying genetic sequence, okay? Now, that's kind of interesting because every cell, really most cells have very different functions. So with the same underlying genetic sequence, what causes a liver cell to be a liver cell, and a hair cell to be a hair cell, and a brain cell to be a brain cell if they have the same genetic code in all of those cells? Well, that's where epigenetics comes in. It actually packages away certain parts of that genetic code that are not needed for liver cells, for example, and makes the other parts of that genetic code that that liver cell does need readily available for the cell. And so fundamentally, this epigenetic modification actually exists in order to, again, regulate that cell-specific function. And so what becomes really interesting is not only does epigenetics function to regulate cell-specific function in order to turn certain genes on and certain genes off that are needed for that specific cell, but it can also turn genes on and off for other reasons, okay? And so epigenetic modifications can be altered by that prenatal environment, which can result in alterations in economically relevant traits in beef cattle, which is where we come in as having an interest in this area as beef cattle producers. So what kinds of things can cause these alterations to that maternal environment, thereby alterations to the fetal environment, thereby actually changing the epigenetic profile of that offspring? So we know that things such as nutrition, so again, all of these can be positive or negative. Now, nutrition can impact the status of the dam, drought, transportation stress, predation, herbicides and pesticides. All of these things can cause changes in the mother's body that can impact that developing fetus. And then the response of that fetus is changing epigenetic codes within the cells of that fetus. And so interestingly, we know that the body, the mammal really body is most susceptible to epigenetic change during fetal and early life development. So the older that animal gets, the less susceptible it is to changing its epigenetic code. So that means that this developing fetus is really susceptible during that fetal development period to program the characteristics that it is going to have after it's born, which is going to impact that animal throughout its life. So that's where we, again, come into really wanting to kind of understand this process, because as some of my colleagues like to say, health and production of beef cattle starts in utero. It starts in the womb. So the research that we have going on really focuses on nutrition. Now, Kentucky 31 tall fescue is a very relevant forage here in Arkansas, in many parts of Arkansas. Now, we know that endophyte-infected Kentucky 31 tall fescue, often called toxic fescue, is the most utilized cool season forage, not only in Arkansas, but the southeastern United States. Toxic fescue does contain a fungal endophyte. And that fungal endophyte actually has a symbiotic relationship with the plant itself that actually helps it be resistant to heat, pest, and drought stress. And although toxic fescue does possess these positive attributes that make it a very hardy and resilient forage type, these ergot alkaloids are produced by that endophyte fungus and have negative impacts on growth, reproduction, as well as overall health and immunity when ingested by livestock, which does make it a problem for producers at certain times of the year. So this brings us to the question of my research project in collaboration with the Livestock and Forestry Research Station. So how does toxic fescue ingested by a pregnant dam impact that developing fetus? So this is something we really haven't put a lot of thought in necessarily to this point in time in the scientific literature. And so our overall objectives of this project were to evaluate the influence of that prenatal exposure to toxic fescue on growth performance, metabolism, and immune function in weaned beef calves. And these are all areas that there has been some research that has shown that might be an impact of this prenatal exposure to toxic fescue. So we want to better understand that. And we also want to characterize that relationship of epigenetic modifications with differences in that growth performance, metabolism, and immune function in calves exposed to toxic fescue during gestation, so that we can better understand that relationship, so that we can better control the outcome of those offspring. And our hypothesis is that epigenetic modifications are in fact associated with differences in growth performance, metabolism, and immune function, and can provide insight into the physiological processes that are controlling those known negative effects of prenatal toxic fescue exposure. So I do want to start by telling you just a little bit about this ongoing research that we have. Now, this project is in progress, so we don't have any data that we're ready to share today, but I do want to just share with you where we're at. Pregnant cows were grazed on replicated novel or toxic fescue pastures throughout gestation at the Livestock and Forestry Research Station there in Batesville, Arkansas. Now, from that subsequent calf crop, we harvested white blood cells from blood samples that we collected from those calves, and then we analyzed epigenetic modifications, specifically DNA methylation, in those samples. So currently, we're in the process of measuring growth performance, metabolism, and immune function characteristics in that subset of calves from that overall population from Batesville. So what we did there is we actually took a subset of those animals, specifically all steers, and we brought them to Fayetteville, Arkansas, where we have a facility that we are able to conduct repeated blood sampling by inserting a jugular catheter in the jugular vein of those animals. And so we can take those repeated blood samples so that we can really understand what's going on in the body of that animal. And so we have specific tests that are designed to evaluate metabolism and immune function separately, of course, in those animals. And so we conducted those actual animal data collections in the summer of this year. And we're in the process of conducting the laboratory analysis on that. And now, after we finish our laboratory analysis, the goal is to go back to our white blood cell samples that we collected after birth, that we have epigenetic analysis from, and compare those differences to the differences that we may see in growth, metabolism, and immune function. And so there are actual programs that allow us to take those epigenetic modification information and actually predict how those changes are going to turn genes on and off and change things downstream in the body, thereby changing the characteristics of that offspring. So we will compare those predicted differences with the differences that we actually see in the animal in order to use those as potential indicators or markers of those changes that we see in the animals. And that brings us to our current goals, which are to better understand how prenatal exposure to toxic fescue impacts calf growth potential and health of those weaned beef calves, and also to identify epigenetic markers or identification of things that are going to help us better understand that growth potential, health, and health parameters in cattle whose dams consumed that toxic fescue. And ultimately, our goal is not to suggest that producers should not use toxic fescue, because it is a very relevant forage type here in this region of the United States. However, we do want to eventually identify and test novel therapeutics to improve performance of offspring born to dams consuming toxic fescue during gestation. And we actually have some simultaneous projects that are going on currently to try to start evaluating some of these therapeutics. Now, with that, I would like to acknowledge some very important people that have worked on this project. All of which are actually not listed in this slide, but Mr. Don Hebel, has been a primary collaborator for this project. We've really appreciated all of Dawn's help. Dr. Jeremy Powell, Dr. Charles Looney, Dr. Beth Kegley, Doug Galloway. The students that are primarily using this project for some of their thesis work is Randy Benefield and Sydney Dunkel, both of which are master's students. And funding and support came from the Division of Agriculture, the Department of Animal Science, as well as the Division of Agriculture Animal Health Grant and Division of Agriculture Batesville Matching Funds Grant. I would like to thank you for your time, and I would be happy to answer any questions at this time.